Hello, everyone. This is Yamina from Dr. GPCR, and in collaboration with the 5th GPCR Targeted Drug Discovery Summit, I'm very excited to have with us today Dr. Will Barnes. He's Executive Director and Head of GPCR Biology at Iambic Therapeutics. Will, welcome.
Thank you, Yamina. I'm happy to be here today.
Very excited to have you on. Maybe we could start at the beginning. If you can tell us a little bit about yourself, your role at Iambic, and what drew you to GPCR biology specifically.
Yeah, terrific. Yeah, no, I joined Iambic a couple of years ago now when they expanded their AI-driven platform from oncology into GPCRs and non-oncology programs. And yeah, I've had a long career now in G protein coupled receptors, starting with my PhD work with Lindsay Huff at Albany Medical College. I studied histamine receptors in the CNS, and I never really looked back from there. Went on to do my postdoc with Bob Lefkowitz at Duke University, and then I spent about 20 years now in biotech or pharmaceutical companies, and have always had a GPCR program as part of my portfolio of programs that I've worked on.
But a true GPCR aficionado. So, having been for such a long time in the field and being a true, let's call it, let's be GPCR nerds today, what are some of the things in your work that still surprise you even now?
I think it's really, for me, the plasticity of G protein coupled receptors continues to surprise me. They're not just simple on off switches, but they can be modulated in very many unique ways by agonists and allosteric modulators that leads to signaling through canonical G protein pathways, but also through bias signaling, including arrestin pathways and other pathways. And it continues to marvel me, and also the polypharmacology is continually interesting to pursue. We used to think of polypharmacology as a bad thing, and now we're really understanding that polypharmacology can really be used to our advantage for bringing novel medicines to patients.
Your point. I spend a lot of my time curating content from our masterclass library, from Terry's Corner, and from everything that's being published. And every time I read something, I'm like, wait a minute. When I initially started learning about this, we were told that internalization is the end of the signal. And nowadays, that's no longer the case, which I find really fascinating.
Yeah, absolutely true. And it's
And make,
wonderful how we can use that into our advantage in bringing new medicines. Sometimes you need a prolonged signaling inside the cell and you want to retain that. And sometimes you have to specifically turn off that prolonged signaling while preserving the acute signaling through the G protein.
your point, it's an intriguing time that we live in because we have now the tools to be able to measure the short and the prolonged signal, but also have the ability to make that connection between an outcome and a medicine that we want versus, being able to modulate that signal and what is the outcome for the patients when it comes to that. Wonderful. So, being where you are today, I wanted to also ask you, where do you think AI-driven approaches are actually closing in the gap on GPCR structural complexity, and where are we still pretending we've solved it?
Yeah, no, I think it's a really exciting time in the field. I think where we see AI-driven technologies such as Neuroplexer, which is the program that we hit with the AI-driven platform we have here at Iambic, it really helps us understand sort of the orthosteric versus allosteric binding, and it is helping us uncover, in some cases, cryptic pockets where ligands are binding. I think where we're still seeing it struggle is in the exact ligand pose that you might have within the receptor and its ability to predict that pose. And I think what's really exciting here at Iambic is we're continuing to build sort of physics-based modeling into future generations of neuroplexer. We really want to get to the place where if we model an agonist binding at an orthosteric binding pocket, we really want to be able to see neuroplexer and other programs be able to give you that conformational change that you expect with receptor activity, such as the outward swing of TM6 forming that kink to allow the G proteins to then dock and then signal. And right now there's a disconnect with those models for those sort of orthosteric binding modalities and then sort of that inward conformational change in the GPCR.
You make a great point. I think being able to really evaluate what conformation is stabilized by a specific ligand and how does that affect signaling is key. And being able to train the models on those will help accelerate the discovery of actually making more of those links between conformation and then function.
Yes, absolutely.
I'm excited to see you in person at the end of this month in Boston. And I wanted to ask, is there a talk, a conversation, or a question you're hoping that this meeting will help you think through?
I think the question that I'm intrigued to sort of see addressed is how others are closing the loop and how they're using sort of these structure-based AI models, coupling that with their pharmacology, experimental data, and then using that with their translational models to make these sort of more intact closed loops from identifying the target all the way up to moving development candidates into the clinic to actually help the patients and seeing that improvement also in translation from preclinical models into clinical trials. I think that's another real opportunity for AI and machine learning models in general in the years ahead.
Agree. And to your point, I think the way I think about structure, about AI, about pharmacology is these are different buckets, and our role as scientists is to figure out a way to make these work together in a way that we can very quickly move from one bucket to another and also be able to close the loop and send the information back and say, well, this didn't work, and this is also valuable information. And this meeting, based on the program, is going to have a lot of very interesting talks, to your point, to see how other companies and how other people think about closing this loop and accelerating drug discovery and finding more drugs towards targeting GPCRs.
Yes, I'm looking forward to it.
Is this going to be your first time at this meeting?
It is my first time. Yes, looking forward to it. Absolutely.
Excited. I hope the weather will collaborate. I'm based here in Boston, and it snowed this morning, and then it rained, and now it's sunny. So all seasons in one day.
Well, I did my undergraduate work at Boston University, so used to the weather, and I look forward to visiting.
Looking forward to having you here. I have a couple of more questions. Outside of your own program, what are some of the developments in the field that have gotten your attention right now?
Yeah, I think as I mentioned earlier, I think it's the polypharmacology that really has me interested, and I think the incretin story is now turned into, a great example for the field of fine tuning peptides in the case of the treating obesity for the GLP-1 receptor, GIP receptor, glucagon receptor, being able to fine tune that peptide to have particular activities at each of those receptors to give you ultimately the pharmacology that you desire with a tolerability profile that continues to improve in patients for the treatment of obesity. I think that's really exciting space. And I think it's another opportunity for our AI models to take in a lot of that data. When you're getting experimental data now, not on one receptor, but maybe two or even more receptors, it's the AI models that can really help you see the path forward in your SAR efforts to drive a low molecular weight compound to be able to have that kind of polypharmacology that we've seen has been achieved with those peptides in the incretin story. I think that's a really exciting space.
It's a very exciting space, and it's something that I think about a lot. Whether we want an agonist on GLP, do you want a partial on GIP, do you want, and what are the combinations of these receptor activities or these compounds should have in order to preserve muscle, reduce side effects, but also ultimately go into taking patients into weight loss. And it's I think it's a fascinating story, and it also goes back to how beautiful the GPCR world is.
Yeah, just imagine. I mean, 20 years ago, polypharmacology was almost a dirty word in the drug discovery field. The idea that you would try to hit more than one target with a single drug was really frowned upon, and that's really changed now.
We've learned more, we have more tools, and with AI being able to test more compounds and also see some, to your point, drive SAR by accelerating, finding these little differences in the molecules and how they affect the signaling has definitely been a factor in changing how we think about polypharmacology in general and pharmacology in general.
Absolutely.
Well, I have one last question. If biology hadn't found you, if you were not a scientist, what would you be doing?
Well, I think I would probably still have been a scientist and probably in the physics field someplace. I think all those years of study, I always found myself in the library with my head in a physics book, and I think that's one of the exciting things about my role here at Iambic is there's still that physics component as we think about the structural modeling of GPCRs, getting to think about just the way that a ligand can change the conformation of GPCRs in so many different ways. I think I would have ended up in physics if not biology.
Close enough. And nowadays, these disciplines like computational chemistry, medicinal chemistry, and physics-based methods and pharmacology have to work together in order to use all the methodologies available to us to move drug discovery forward.
Absolutely, yes.
Thank you so much for your time. Don't go anywhere. I really appreciated you giving us a little bit of your time. I look forward to seeing you in person in Boston with the weather cooperating. But if not, I think the room is going to be very warm and very excited to be talking about GPCRs again this year.
Looking forward to it. Thanks, Shamina.
Thank you so much.